AISA has in his basic concepts, which means anti-inflammatory senescence actives, the endothelial target, which is cell adhesion molecules recruiting activated leukocytes. Now, this process is taking place several times a day for immune purposes, and it is not toxic at all to endothelium. It becomes toxic when leukocytes are activated and the process is augmented in rhythm and time. Eventually, cell adhesion molecules can recruit also circulating tumor cells. In a previous work I had done on some glutathione peroxidase mimics, we had noticed that the mimic which was able to perfectly inhibit cell adhesion molecules was also able to inhibit actin polymerization. And I think we should take into account this aspect of the modification of shape of the cell through cytoskeleton, because cytoskeleton is importantly linked to cell signaling. And so to us, anti-inflammatory senescence actives do first target vascular endothelium. Secondly, they do inhibit cell adhesion molecules to be expressed following stimulation by pro-inflammatory agonists. And third, they do reverse the senescent phenotype, which is very interestingly linked to a modification of cell shape. In fact, what you see there is not the product of TNF, it's the product of just cell replication. In the course of time, the cell becomes stiff and it expresses ICAM1 without being stimulated by TNF or other agonists of inflammation. So what we think is interesting in this case is to be able to reverse this specific effect. We had a little screening platform in our University Paris 6 in France, Paris, where I was sitting at the time, and screening some molecules coming from the prestigious Institut de Chimie des Substances Naturelles of the Senescence and of China's Academy of Medical Sciences. So we started to look for a molecule coming out from a plant doing precisely these jobs I have told you. So, there it doesn't come out, doesn't matter. Oops. We started with a tryptorine, which is a triterpene isolated from Triterbium wolfordi hook, a perennial creeping plant indigenous from southern China. What is interesting in this plant? That it is eaten since thousands of years in the soup to prevent rheumatoid arthritis. Now, this work was able to confirm that, in fact, the molecule, which we couldn't see because there is a PC overwhelming organization here, was able perfectly to inhibit adhesive molecules and also selectins, very interestingly. And how do you go back? How do you go? And when we compared it to an anti-inflammatory drug panel, what we can see here, and this I think is a very interesting result, I mean, normally anti-inflammatory drugs do not inhibit adhesive molecules when endothelium is stimulated by TNF, besides ibuprofen, whose job is doing quite well towards VCAM, not so good towards ICAM. So in ICAM, we are sure that none of the drugs we are giving to patients are doing the job of inhibiting adhesive molecule expression. So with the second partner, which was Institut de Substances Naturelles, we, through a bio-guided selection, we were able to identify 4 molecules out of 2,000 tested, originating from some 100 plants extracts. We were able to file a patent, and the molecule I would like to talk about today with you and just present some results is anti-inflammatory senescence active 5203L, And L is limonen, the major component of orange peel oil, but also found, interestingly, in breast milk. It is a prototype of monoterpenes in carcinogenesis studies, and it is interesting not only by itself, but also by its metabolites, which are inhibitors of isoprenylation enzymes. And endothelial signaling is quite implicated by proteins of the raw GTPase family, and everybody knows that the activation of these proteins requires a post-translational isoprenylation. So the major metabolites, the other reason we have chosen this molecule and not the others, is that we were quite assured about a pronounced chemotherapeutic activity and minimal toxicity in preclinical studies. It is known that in phase 1 clinical trials, this molecule is not toxic, although it's not having a big success to make cancer stop. It is not toxic, and pharmacokinetic studies show that it is tolerated. So it was in our hands perfectly able at all replicative stages to stop adhesive molecule expression following TNF-alpha stimulation. And it was at 24 hours not toxic at all, and at very low concentration. Sorry. We then had the problem to pass to animal studies. And I thought, no, I will not do that to now start to shoot rats with this stuff. And I thought I would do another experiment, which was to acknowledge the link between stress and inflammation, which we know from literature since so many years. Hans Schelle in 1936 already was stressing that stress is a general alarm reaction, and it might be compared to other general defense systems, such as inflammation. Myself, I had given a review some years earlier establishing the link through aging and endothelium, and the link was precisely stress. So we used a functional observation battery of 44 tests, and we measured behavioral effects, neurological effects, and physiological effects, which are listed up here. But very precisely, I would like to state that the stress situation was a very low stress situation, such as maternal separation or academical stress, the one I am experiencing at the moment. Or, for instance, putting somebody in the middle of a room and putting all the sound away. So those were rats put in this very slow stress, and given obviously the molecules and AISA therapeutic molecules and the metabolites. So what we have checked first, because to me there's an impressive link between motor functions and mood alterations, we have looked at the first simple thing, which is spontaneous activity. And spontaneous activity was impressively higher, maintained, stable, compared to the control, to the stress control. So the animal felt free somehow. Square crossed performance test. Wire maneuver. The wire maneuver is a maneuver in which the animal must be able to keep going on a metal wire. And the negative geotaxis means just that if I return the animal, it goes back to its normal position without difficulty. In red, you see the effect of limonen. What about pain response and mood modulation? The pain response is here, and you see quite interestingly how immediately there is a very important response to the molecule. And freezing means that the animal is completely paralyzed by anxiety.